Thank you. It's a pleasure to be here. Thank you for the introduction. I'm also part of the Recover Pediatric cohort and lead the long COVID clinic at CHLA. Okay, so I didn't go too much into the long COVID description because I am speaking to a group of people heavily invested into long COVID. But I just want to make one distinction, which is slightly different between adults and children, that in children there are two types of sequelae that occur after COVID infection. One is called multisystem inflammatory syndrome in children, which is MISC, and the other is long COVID as we see it in adults. And the mechanisms underlying why certain patients develop COVID-19 sequelae, whereas others do not, is poorly understood. And I think that has been a question that I have been trying to investigate from the beginning of the pandemic when we first saw MISC. So investigating the genetic predisposition may actually illuminate underlying disease mechanisms, the reason for susceptibility to sequelae, and inform targeted therapeutic approaches. So the morning session for the mechanistic talks was really awesome, and I was so inspired and excited to find tie-ins with what I have found as well. So just as a background, we looked first into whether there is genetic predisposition for COVID-19. And there are genome-wide association studies, or GWAS, which have found strong correlation between specific loci and the COVID-19 susceptibility and severity, which you heard a talk this morning about the loci which could be important, like the ABO and the ACE2 pathogenic variants. These findings support the hypothesis that both acute COVID-19 and by extension, long COVID are significantly influenced by host genomic factors. So looking at long COVID in particular, this figure was also there this morning, and the speaker emphasized what they found within the long COVID group. And GWAS found strong associations between the FOXP4 gene and long COVID incidence. And in this group, like already emphasized, this was thought to be primarily by its function in lung function and lung physiology. There's another study which focused on long COVID patients, particularly with the severe fatigue phenotype. And they identified multiple genes enriched in this group, and they did a pathway analysis and found that were predominantly related to neurologic and cardiometabolic pathways. So most of the studies looking at genetic susceptibility include or involve adult cohorts. And I would argue that studying a pediatric cohort for genetic susceptibility has more value. Disease status is more likely to be linked with genetic predisposition rather than accumulated environmental factors. And there is reduced confounding from the cumulative environmental stressors that accumulate in adults. So going into the approach, we used the recover samples for this testing. And you will see that we had three cohorts: PASC positive, that's long COVID positive, MISC, and PASC negative. PASC negative is patients who had COVID but did not develop long COVID. And this distinction was made using the long COVID research index that's part of the recover cohort. And as you see here, we had a sizable number of patients, PASC negative N of 98, PASC positive 102. MISC is a smaller sample. That's because due to some data issues, we have not yet been able to include the whole cohort. And as you see, the age, there was a good distribution of age, and the majority of the patients were between 13 to 25 years old, which is kind of the major breakdown in the whole recover pediatric cohort as well. There was a slight male predominance in the samples that we had, and we had a good breakdown of races. The majority were white or Hispanic. The sequencing was done at Children's Hospital Los Angeles, and all of these samples that were tested were saliva samples. So the methods that we used the Illumina Dragon to call the variants. We did single nucleotide and small insertion deletion variants were classified using the ACMG classification. And if you look here, the figure actually shows the ACMG classification, which classifies the variants from benign to pathogenic. So we used pathogenic or likely pathogenic variants in genes associated with known autosomal dominant or X-linked disorders and filtered them for analysis. The frequency of pathogenic variants was compared between both cohorts and the PASC negative cohorts. So we found 151 pathogenic variants across all the cohorts. And the average number of pathogenic variants per sample, if you see, there's not a huge difference between the three groups. The PASC positive was 1.5, whereas the PASC negative was 1.28, suggesting that the total number of variants by itself is probably not important. It's probably what the variants, which gene is affected. Then the next thing we did was we grouped this pathogenic variants by functional group to see if there was a specific functional group that stood out in any of these cohorts. And what we found was, so this figure shows the MISC cohort and the long COVID cohort. So there was a distinct enrichment pattern between the two sequelae. So if you look at MISC, organelle and trafficking and other specialized functions stood out as the pathways that could be important. And within long COVID, the two really that stand out the most is signal transduction and mitochondrial function. So some of these things have been mentioned earlier in the literature. Metabolic pathways are widely known to be disrupted in long COVID patients. Mitochondrial function has been a potential target for therapeutics and has been identified as a potential cause for long COVID. And again, that's also something that came up in the discussion this morning. I was very excited to hear about the mitokines, which was, I think, in Dominic's work, stand out as one of the things that was different in long COVID. And the novel findings that we have are the signal transduction pathways standing out specifically in long COVID. While they've been noted previously in long COVID studies, they have not been extensively investigated as a primary focus. So the next thing we did, we looked at the individual genes that were enriched. And again, compared the normalized counts of these pathogenic variants compared with the control group. And you see the names of all these genes that stand out. I would like to highlight in particular within the long COVID, the polymerase gamma or the POLG gene, which was, as you can see, really prominent in the long COVID group. And the MISC group, on the other hand, had FLG and VWF, which has a role in hemostasis, and we know that these patients have significant disorders in hemostasis when they first present to us. So these were what stood out distinctly within these two groups. And looking at these individual genes, when we looked at POLG, for example, it's been linked to long COVID through mitochondrial dysfunction. The other thing that was the gene that stood out is MEFV. This is linked with post-acute COVID-19 in inflammatory symptoms. I realize I'm a little short on time. Okay, and VWF is increased in patients with MISC. So if you look at the individual gene enrichment, POLG or polymerase gamma, it's an essential component of mitochondrial DNA polymerase. It's responsible for replication, proofreading, and DNA repair. And disruption of the POLG can result in overall mitochondrial dysfunction and accumulation of mitochondrial mutations. So if you look at this schematic, so compared to a normal person, I mean, someone who's not affected by long COVID or who has this POLG variant, over time, due to the impaired mitochondrial DNA replication, mutations in the mitochondria accumulate. This leads to dysfunctional mitochondrial function and energy depletion. And energy depletion or the altered energetics may be one of the leading causes of the symptoms, which include fatigue, exercise intolerance, PEM, and even the neurological defects. The other individual gene that we looked into was MEFV. MEFV produces pyrin and has a role in inflammasome activation. It's a variant that's identified in FMF, or familial Mediterranean fever. And there are actually reports of long COVID with MEFV variants. So if patients with long COVID present with recurrent or persistent fevers, this is not a usual phenotype, but if they do, MEFV's variant is one of the things that should be looked into, because there is a treatment, colchicine works pretty well in treating this condition. So to conclude, these findings validate the approach of comparing pathogenic variants between the disease cohorts. Our results demonstrate findings consistent with previous research, while also providing novel genetic insights. And both functional gene groupings and individual pathogenic variants identified here have potential avenues for mechanistic research and therapeutic development in the future. This is a wordy slide just to say that for the future, what we really want to do is look at DNA methylation and also further investigate the mitochondrial mutations. So we are working on both of these things, and actually the early signals, especially with the mitochondrial mutations, is very impressive. So hopefully, once we are done with that, maybe I can present it at the next meeting. And one of the, during thinking of all these findings, one of the things that we were thinking of was the GDF15, which was also mentioned in a talk this morning as a potential marker which stood different between long COVID and other patients. So yeah, very exciting, and we look forward to finding more or delving more into these findings. I want to thank my study team, and funding-wise NIH, as well as Recover, and the Research Institute at Children's Hospital, Los Angeles. Thank you.